Hey guys, let's begin with another important topic today. So, warm welcome from the Spice Up Your Biology. Today, we're going to talk about the protein targeting or protein sorting to the nucleus. How exactly the protein which is being synthesized in the cytosol. We know the place where the protein will be synthesized is the cytosol. So, how exactly the protein which is being synthesized into the cytosol will enter into the nucleus? Will it have some address or not, or something else which is going to happen? Let's go into the basics of that. Just look at the cell. We can define it as a compartment within compartment. How this compartment, they interact with each other, how the proteins that are going to be sorted from the one place to another, especially from the cytosol to the different cell organ. But today will be very specific to the nucleus. Let's talk about the one. So how exactly the intracellular compartments, and in that the protein targeting is going to take place. First, we'll talk about the protein sorting to the nucleus. Cell, as you see, it is like compartment within the compartment, as I told you, and they have the different molecules. Look at the membrane, which is the partitioning one cell organelle to the other. And if you look at the basic property of the membrane, so membrane do have a property. It will not allow the hydrophobic molecules to go in unless it has a protein for it. If it has the protein through which this molecule can pass, it will go. Otherwise, it will not be able to go. Unlike the lipid or the lipid soluble molecule like the steroid, they can easily pass through the cell. So whenever something which is going to enter into those compartments, they require to go through the some kind of transporter proteins, and they should always recognize some kind of signal present on those molecules one way or the other. So the major cell organelles that a normal cell have, I'm talking about the animal cell. For example, the nucleus, cytosol, endoplasmic reticulum, Golgi apparatus, mitochondria, chloroplast, lysosome, endosomes, and peroxisomes. But today, as I told you, we are going to be very specific about the nucleus. But we cannot, go into the detail of this topic without, making it a bit journal, so that you can understand, we can have an insight into the concept, and then we can define it to the nucleus. Look at, these are the cell organelles which are present within the cell, and they have a particular way of their origin. We'll talk about it. There will be three ways that transportation can occur. One is a gated transport. That means we have some gated channel, which is going to be topologically equivalent. That means the exterior and the interior is going to be similar. Then there'll be transmembrane transport through the protein translocators, and third one is going to be the vesicular transport. And it has been shown into the different. For example, the red color, which is the gated transport, which is very similar to the what we are talking about. The blue one, which is for the transmembrane protein, and the green one from in the form of vesicle from the one compartment, especially the ER, plasma membrane, Golgi apparatus, they will play important role there. And these are different compartments within the cell where these pathways are going to be happening. Whenever the transport occurs, this transport is going to be guided by some sorting signals, which is going to be recognized by the transported protein. And these signals are always going to be present on the transported protein. And the receptor is going to be complementary, which is going to recognize those sorting signals. And there'll be two kinds of sorting signals which can be present on the protein or the cargo, which is moving from one place to another. And they may have a continuous sequence of the 15 to 60 amino acid. Sometimes they're going to be removed from the finished protein, or they may be the part of the finished protein. That could be one way. Or maybe the signal patches, which help the protein to be folded into a 3D structure. And amino acids which are distance, they might come together in the folded product. Now there's a certain signal. Again, one thing that you have to ensure that these signals can be present on the either side of the protein protein has the two terminus, N terminus and the C terminus. So those signals can be present on the either the N terminus or the C terminus, or it could be on the either side. For example, anything which is coming into the nucleus will have these signals which contain the two proline. There'll be lysine, arginine, that means the basic amino acid, and it will end up with the valine. It could be on the either side. Now, any protein which is moving out of the nucleus, they will have a signal which is going to be rich in the hydrophobic amino acid. For example, the leucine, alanine, leucine, there'll be some positively charged amino acid and some negatively charged amino acid, but largely it is going to have the hydrophobic amino acids. Similarly, the any protein which is entering into the mitochondria have the patches of the arginine and the lysine, which are positively charged, along with the some of the other hydrophobic amino acid and some other amino acid. Similarly, for the plastid, we have the certain important, like which is shown in the blue. Serine residues are going to be very important, which is present at the regular intervals. Similarly, for peroxisome, they have a signal which is present largely on the C terminus. You can see that there'll be three amino acids: serine, lysine, and the leucine. Again, if any protein goes into the endoplasmic reticulum, they will have largely the hydrophobic amino acids. And anything which is returning to the ER will have the lysine, aspartate, glutamate, and leucine KDL signal on the C terminus. These are just some general concepts. We'll go detail into the nucleus. Now, as I told you, they have the 3D patches or the signal patches. These are the proteins which might go to the two places. One is a nucleus, another is going to be lysosome. Whereas for other places, we have the signals present on the N terminus or in the C terminus that already we have discussed. Except that, in the case of peroxisome, they have the three amino acids that we already have discussed about, which can be present on the C terminus. Now, the salting signals, they recognize the complementary salting receptors. It's like, a kind of papers that requires to get into the some kind of, the warehouses. So they'll be checked, and once it is checked, the cargo, which is the moving protein, can be unloaded, and those can be used time and again. They will be having the reusable kind of thing, and they can function catalytically. Now, if you look at the nucleus structure, so you have the nuclear envelope. This is the only envelope, all the membrane bound structure, which is not continuous. We have the outer membrane, we have the inner membrane, and they have a lot of perforation or discontinuity due to the big pores, because the nuclear pore or the nuclear pore complexes, which are the places through which I told you in the beginning, there's a gated kind of transport. That means it will come through a gate. Whereas the other places like the mitochondria, endoplasmic reticulum, plastids, they have a transmembrane transport that you have to be very careful about. Now, the nuclear pore complexes, a brief, we look at, like, this will give you a feeling that we have in the basketball court, the basket through which the ball can be put in. So we have, those structures, the octagonal structures. They have a lot of fibrils which can be seen over here. And at the bottom of the inner envelope, inner membrane, we have a green color structure, which is made up of nuclear lamina, which is kind of intermediate filament. The mass of nuclear pore complexes around 125 million, and they are made up of 50 different proteins, and they are going to be arranged, as I told you, in the form of octagon. And they have, the number can vary, usually from 3,000 to 4,000 in the mammalian cell, and they may have, more than one aqueous channel through which molecule of the smaller size can easily pass. So usually around 5 kD they can pass, and beyond that it will not find, that easier to go through. And other proteins, I mean, the anything which is coming through the receptor protein will require some kind of energy. That means that transport is going to be active transport. Look at this, the pore I was talking about. Smaller protein up to the 5 kD of the size, can go into without any kind of energy input. But bigger proteins, they require the energy. That means they require the input of the ATP in order to go. Now, whichever the protein that is going to enter into the nucleus through the nuclear pore complexes, they have a signal we call it as a NLS, nuclear localizing signal, generally comprised of the two short sequences, which is rich in the positively charged amino acid like the lysine and arginine, and they can be located anywhere, unlike the other cases where it can be present on the C terminus or it can be present on the N terminus. And they've thought to form the loops. I told you those patches will help in the folding of the protein to get the 3D structure. Again, one more thing that you have to keep in the mind, the protein can enter into a cell organelle either in the completely folded form or it will go into the unfolded form. There inside this is going to be folded. Okay, now once it enters through those pores, and protein will always enter into the nucleus in the completely folded form, and this is an active process. I told you in the beginning, this requires ATP. Now that the few players which is playing a very important role between the cytosol to the nucleus, for example, the importins. These are the cytosolic receptor of the protein, which is going to recognize the NLS, the nuclear localizing signal on the cargo protein, and it will bind to it. The second one is a nuclear export receptor, which is bind to the macromolecule to be exported from the nucleus. And there'll be adapter, which is playing an important role, which requires to bind the target protein to the nuclear receptor. Then we require one monomeric G protein that I already have discussed in the G protein lecture, Ran, which is a cytosolic GTP and GDP binding protein that can switch between the active form to the inactive form. We'll talk about in a while why it is so. Then we have the fibrils, which is the inner part of the nuclear pore complexes, which has the FG repeats. That means they are going to have the phenylalanine and the glycine repeats, which helps to recognize the protein which is entering or moving out of the nuclear pore complexes. Even I told you while talking about the export of the messenger RNA, there also they require to scan through the protein bound to the messenger RNA in order to make a move. And the movement of those complexes is going to hop into the nucleus. You can see in this diagram, I'll just make sure how exactly it is going to work. Suppose this is the cargo protein which is in the folded form. This is the exposed part of the NLS. Importin will come and bind to it. And once the binding happens, then it is going to bind to the nuclear pore complexes, and there it will go. You can see over here there will be GTP which is going to be utilized, that is present on the Ran. Similarly, how exactly this process occurs? So we have a molecular switch, that is a Ran-GTPase. This drives the directional transport in the appropriate direction. The conversion between the GTP and GDP bound state mediated by the Ran specific regulatory protein, we call it as a gap. So gap converts Ran-GTP to the Ran-GDP via the GTP hydrolysis, and GEF, like any other monomeric G protein, that will exchange the GDP with the GTP. Now, where exactly these two proteins are going to be functioning? For example, the Ran gap is going to work in the cytosol so that it can convert Ran into the Ran-GDP into the cytosol, whereas Ran-GEF is going to be active into the nucleus, where it converts Ran-GDP into the Ran-GTP by exchanging the GDP with the GTP.